We're going to go ahead and get started. We're about 40 minutes behind schedule, so I'd ask all the speakers as they come up to try to stick to 15 or 20 minutes maximum. So I'm going to shift gears a little bit this morning and talk about cardiac regeneration and cellular reprogramming as opposed to stem cell delivery. I'm the DeBakey Bard Chairman of Surgery at Baylor College of Medicine in Houston. And of course, at Baylor, with the tradition of Dr. DeBakey, understanding how to repair and fix the heart is of course a significant priority. In the United States, heart disease, of course, remains a very significant problem, and basically the numbers that are existent in the United States can essentially be doubled worldwide. So in the United States, about 5 million Americans have heart failure. About 2% of the population. Current medical and surgical therapies, many of which actually were invented at Baylor, heart transplant, first in the United States, the artificial heart and the like, are available, but of course are limited in both in terms of the availability of the artificial heart, the cost, the morbidity, etc. So clearly an additional therapy is needed. By some estimates, in the United States alone, there are at least 500,000 to 1 million who need an alternative to current therapies of artificial heart or heart transplant, and again, those numbers can be doubled in the Western world. Most people, when they think of end-stage or severe disease, think, of course, of cancer. But interestingly, patients with end-stage heart disease, in fact, carry the same mortality risk, in many cases even worse than patients with end-stage cancer disease. So the 5-year mortality for patients with end-stage heart disease is 60%, again, exceeding most cancers. And even with artificial heart or VAD, ventricular assist device, or transplant, the 2 to 3 year mortality is 20 to 30%. So clearly, an alternative is indicated. Now, most of the work that's gone on in the last 5 or 10 years, and we're going to hear about much of this this morning, involves cardiac stem cell therapy. So taking stem cells, iPS cells, other types of primordial cells, administering them into the heart muscle, into the area of infarction or scar tissue, with a sense that this will cause regeneration of this scar back into functional muscle. What I would argue, and what we're going to talk about in the next 15 minutes, is that implanting exogenous cells, as you can see at the bottom left over here, into an island of scar tissue is unlikely to be successful. It's very difficult, we believe, for these cells to become integrated into the host myocardium. And certainly, and more importantly, those cells are unlikely to survive without an adequate blood supply. This is certainly the reason why the original cells died to begin with. And what I'm going to talk about this morning is a completely different therapy as opposed to implanting exogenous cells, outside cells, into this area of scar tissue, really very much a desert of inadequate blood flow. We're going to talk about reprogramming the endogenous in situ fibroblasts back into cardiomyocytes. So literally something that would have been unthinkable but 5 years or so ago, but with the Nobel Prize winning work of Yamanaka, showing that adult cells can be turned back into iPS cells, the door really was open for all sorts of interventions. Let me digress and talk about my background in terms of this area of work, gene therapy. Back in the late 1990s, when I was at Cornell collaborating with my partner Ron Crystal, we were actually the first to inject an adenovirus or any type of virus into the human heart in the United States. One other investigator in Finland had done work prior to us. And the goal was to induce a new blood supply to the heart for patients with end-stage coronary disease, so such severe disease that there was no place for I as a surgeon or my colleagues to sew a bypass, or our cardiology colleagues to put a stent or an angioplasty. And so the concept was essentially we're going to take VEGF, we were going to put it into an adenovirus, as seen at the top left, use the adenovirus basically as a delivery vector to deliver the VEGF to the human heart. And this obviously involved 10 to 20 years worth of work, but in a long story short, what we were able to show, and this is an example in a pig model, that delivering VEGF using an adenovirus would allow new blood supply to develop. And again, seen on the bottom right, the first work was done in the late 1990s. In 1997, the first time a virus had been injected into the human heart. So these trials advanced along, and this is reflective of a multi-center study that had been formed in Canada. On the left, the endpoint was time on the treadmill, time to develop EKG changes. And what you can see here is that in a medical control group, which is the gray, versus the VEGF adenovirus group, as early as 6 months after therapy, we found a statistically significant improvement in exercise treadmill, a data point that we know was appropriate for FDA approval of this and other drugs. As shown on the left, there was tremendous amount of enthusiasm about this therapy. This was one of our patients who was actually a reporter for Time magazine. He had had a number of bypasses, had end-stage angina and coronary disease, and literally could not function in the office. And he writes in this article in Time magazine that he got up after the gene therapy. Again, all we did was inject a virus into the heart carrying the VEGF gene. He was able to go back, play squash and the like, and he subsequently wrote a letter to our Vice President, Dick Cheney, who also had heart disease, ultimately had a heart transplant, and said, well, you know, these guys in New York are doing great work with VEGF and gene therapy, and maybe you should check it out, and maybe he should have. I wouldn't have ended up with someone else's heart. But in any case, tremendous amount of enthusiasm. This is in the late 1990s. So why are we sitting here in 2015 and there's only one gene therapy that's been approved worldwide? What happened? And I would argue quite simply that it was a combination of some bad science, and we don't really have enough time to talk about that, and also some heightened enthusiasm and sort of hyperbolic expectations, and then the crashing reality of, again, poorly designed trials. And as depicted here in 1999, Jesse Gelsinger was undergoing a clinical trial at University of Pennsylvania and quite frankly probably received a massive overdose of adenovirus inadvertently, died suddenly, that put a moratorium on gene therapy according by the FDA, and that really derailed gene therapy in the United States for at least a decade. And only in the last 5 or 6 years, as we all know here, has gene therapy regained its credibility, and again, with the good science, some excellent trials are going on around the United States. In terms of our own interest in the field, what happened is after we did our original phase 1 trial at Cornell, many years passed, we began to lose track of our patients, but then about 10 years after the trial, one after another patient were coming back, reapproached us, said, hey, Dr. Rosengart, I'm still doing well, just wanted to let you know I was still alive. And that prompted us to look at the entire cohort of patients we had treated. And we found that our median survival, in fact, exceeded what we had expected. And in a number of patients, as seen on the right, we saw that same collateral network of new blood vessel growth, at least anecdotally. It was suggestive that something was going on. Again, this is 10 years after therapy. And that led us to launch into a trial. This, again, is our results compared to our control group, showing a significant improvement in survival, or an encouraging data suggesting an improvement in survival. What we went on and did in the last 3 years is go back to the FDA and said we wanted to go create a new gene therapy trial. In this case, we're using a VEGF isoform genomic that contains all 3 isoforms of VEGF, 121, 165, 189. We found this to be about 10 to 100 times more potent than the VEGF adenovirus that we used back in the late 1990s. And with the FDA approval, we are in the next couple of months, we started a small company called Xylacor to fund the trial. We're going to do a placebo-controlled trial, randomizing patients to VEGF therapy with adenovirus versus a null virus placebo control for patients with end-stage coronary disease. Now, interestingly, even though our animal data looked very encouraging, the one variable that we didn't know is how many patients there were still with end-stage coronary disease who would potentially be a candidate for this therapy. And interestingly, as we're getting ready to embark upon the trial, as we've begun to speak to our cardiology colleagues, there, in fact, appear to be a great number of patients, interestingly, despite advances in drug therapy, who do have advanced coronary disease, do have significant angina, who our cardiologists are actually looking for therapies, as hopefully we are proposing for treating these patients. And in fact, the first patient I encountered actually had normal heart function, but as such debilitating angina was actually being placed as a candidate for heart transplant. So we actually rescued that patient off the heart transplant list, and we'll hopefully be treating the patient in the near future. So let me shift gears. Now that I've given what I hope is some data suggesting that we can actually grow new blood vessels in the heart, and talk about the concept of how to treat an infarction. Ischemia, of course, suggests that there is a living heart muscle, in which case providing a new blood supply is an appropriate and adequate therapy. However, once the patient's gone on to infarction, again, patients with end-stage heart failure, just giving the heart a new blood supply is not going to improve heart function. And again, as suggested here, just administering exogenous stem cells, we argue, may not be an ideal therapy. There are problems about adequate exogenous cell delivery. We know about 98% of those cells don't survive intact and get retained. There's a question about how well these exogenous cells sitting there are going to become integrated into the host myocardium. And of course, there are the issues about arrhythmias and the like. Now, Yamanaka, back in 2006, 2007, as we all know, demonstrated that you could take the Yamanaka factors, OX, KLF4, and c-MYC, and take adult somatic cells, turn them back into embryonic stem cells, or what he called iPS cells, that he won the Nobel Prize for 3 or 4 years ago. But even iPS cells have problems. They are potentially immunogenic because they've been altered, and they also have tumorigenicity problems. But interestingly, because of Yamanaka's work, a scientist at UCSF named Deepak Srivastava said, well, if you can take one cell, take it from an adult cell type and change it to an iPS or an embryonic stem cell, perhaps you could even go further, identify the transcription factors that are expressed in embryonic development from embryonic cell to a cardiogenic shell, take those same take those same transcription factors, which appear to drive cardiac cell differentiation, and administer them into another somatic cell, and take that somatic cell, turn it into a cardiomyocyte. And what Srivastava did is he took 3 of these transcription factors, GATA4, MEF2C, and TBX5, administered them to fibroblasts using a retrovirus. We used a lentivirus, and see whether those transcription factors would dedifferentiate or transdifferentiate those fibroblasts into what he called induced cardiomyocytes, or ICMs. Srivastava and we subsequently looked at these analyses by FACS staining for appropriate cardiac markers, in this example looking at cardiac troponin T. And what you can see is across the top row, we see expression of the cardiac troponin T in the cells that have been treated with GMT, or GATA4, MEF2C, or TBX5, versus the control fibroblasts across the bottom, where you see no such expression of cardiac troponin T. And in fact, when we try to duplicate Srivastava's work, we received or obtained very similar results. When we looked at CTNT, myosin heavy chain 7, and even alpha sarcomeric actin, all markers specifically of cardiomyocytes, we found expression in the ICM or the GMT-treated cells without expression in the control cells. So this raised a very interesting possibility. If you could take 3 genes, 3 transcription factors, inject them into scar tissue, and take those scar fibroblasts and convert them or transdifferentiate them into cardiomyocytes, this would abrogate the need completely for injecting exogenous cells, providing a new scaffolding, new nature for or environment, microenvironment for these cells to survive, because you literally are creating this as an in situ cellular reprogramming or transdifferentiation strategy, again abrogating the need for exogenous cell delivery. Now, again, importantly, and we believe this is something that has been missed by many, many clinical trials and investigations. We know that when you inject exogenous cells into the area of infarction, nearly 98% of those cells will not be retained. In fact, most die within 48 hours. So this hypothesis led us to hypothesize that if you're going to inject cells or transdifferentiate cells in a hostile environment of a myocardial scar, you first need to supply a blood supply to ensure that ischemic death and apoptosis of those redifferentiated or transdifferentiated cells don't occur. So in our experiments, what we did is we take a rat coronary ligation model. We created infarction. Since we know how to grow new blood vessels, we took and first treated those cells with adenovirus encoding for VEGF. We waited 3 weeks before we induced a reprogramming, delivered our GMT versus a control virus, and then waited 4 weeks to look at outcomes. If I can try to play this. No, okay, so it's not going to play, but basically across the top you see the control hearts, and the bottom the treated hearts. As seen on the left, there's a very large bland scar in the control hearts. However, when we stain for myosin heavy chain 7, a marker of cardiomyocytes, we see these islands and strands of what appear to be reprogrammed cardiomyocytes in the treated heart versus the control hearts. Perhaps more dramatically, in the control hearts, the blue is fibrosis, and we can see that in the control hearts there's a large area of fibrosis and even aneurysm formation versus the treated hearts where there's a much significant decrease in there. And as we'll see in a second, in terms of function, similar results. So quantifying these results across a series of animals, this was published in 2013 in the Journal of American Heart Association. We can see a significant improvement in the induced cardiomyocyte density versus the control animals. Similarly, the amount of fibrosis decreases by nearly half. Really dramatic results. Actually makes us think that there are other processes going on. And finally, looking at function, we found that in the animals that were pre-treated with VEGF plus the GMT reprogramming versus various controls, we found a significant improvement in ejection fraction measured by echocardiography versus these controls. Finally, in another series of experiments, whereas we realized that if we had to administer the GMT with various viruses, each encoding for one single of these transcription factors, we thought if we combine these transcription factors into a single virus, this is a lentivirus, we would enhance the efficiency because the target cells would not have to be targeted by 3 separate genes and viruses. Simply one hit of a virus would be effective. And when we repeated these experiments, looking at our controls and our single viruses versus what we now call our triplet GMT virus, we found that we were able to improve ejection fraction by 30% compared to these controls. These are very, very significant results in the setting of such animal studies, reconfirming our prior work, and again, suggesting significant hope and promise for this, what we think is a fairly exciting innovation in terms of addressing the problem of heart failure. So in conclusion, we think that gene therapy is the good news. It's back, receiving tremendous attention, again, in the scientific and the investor community as well. We think that VEGF pre-treatment of SCAR is critical for these SCAR therapies to be effective. And finally, we think that a cardiac cellular reprogramming, again, offers an interesting and exciting possibility in terms of treating patients with end-stage heart failure compared to conventional therapies. So this is our lab group at Baylor College of Medicine. Has done a tremendous amount of this work. And again, we hope to bring new developments to the community in the near future. Thank you very much.